I'm going to tell you something about what is called translating basic research to experimental medicine. And this is a title that has a lot of sound bites in it. So what that means is that it has all these really fashionable words at the moment. And the fashionable word is what translating in this context means. Basic research is something that we come across a lot. And then experimental medicine is, again, a term that has started coining relatively recently. And therefore, just like anything else that you need to do, you first need to clarify what are the terms that you come across. What it is that they mean. What is it that we mean by basic research? Basic research is also called pure research. And that what it means is it's not applied. So you're not researching around a subject that would allow you to solve a very specific applied problem right away. So you're not researching in order to find out how to make this roof waterproof or something like that. So you're aimed to, your work is aimed at expanding knowledge altogether and understanding the world better. So you're just going to do very careful study of a whatever phenomena, and it's driven by curiosity primarily. Experimental medicine, on the other side, refers to a branch of medicine where we carry out investigations in human beings, and this may be in a whole body or in a person, or samples provided by these people. And this is in order to understand health and disease, or to test new discoveries, to test new treatments. And experimental medicine precedes what what you probably know more about, and it's called late-phase clinical trials. So when we say how to translate basic research to experimental medicine, we really refer to this process of discovery from just working on something rather basic, understanding some molecular processes, understanding some cellular processes, and then coming across a discovery or enhancing the knowledge of a health or a disease state, which then clicks and all of a sudden becomes a possibility to apply it to treatment and to apply it to medicine. And this can be in a fashion that allows us to understand better our health state, or it can be in a fashion that allows us to treat the disease. As you can imagine, in a country like ours, where we have a lot of basic research in the world, recognized and leading basic researchers work in our country, there is a lot of pressure on somehow capitalizing on that body of work generated by basic research and translating that into direct impact to society. So that's what we mean by translating and by translational medicine. Once you made that leap, then you will come to the point of looking into carrying out clinical trials. And clinical trials are done such that we would firstly ask two questions. Is new medicine working, and is it safe? Because this is what we need to know. It's all good in us, in some scientists coming up with the idea of a new drug, but this drug we have to show will work, and secondly we have to show it will not kill you on the way, so it has to be actually really safe. And there are several phases of clinical trials. In the first phase, we will primarily test for the safety of the treatment, and this will be done on a small number of health volunteers. Okay? And what I can tell you that about 50% of drug leads or molecules that we were initially hoping would be potential drugs would fail at that stage. They will show that they're too toxic, that they cannot be tolerated too well, and so on. So they will fail at that stage, and we will not proceed further at that point. In the second phase, phase 2, clinical trials, we will be looking at a larger cohort of people that are ill, and at that point we will assess the efficacy in short term. Yeah? So we will be looking at, I don't know, depending on the disease, hundreds up to 1,000 of patients, but really depending on the disease that you're looking at. And by first establishing the level of tolerance that the healthy individuals will have, in this phase, we will be using those levels of medication in order to test for their efficacy. Once these are carried out, and at this stage, probably about 30 to 40% of drugs will make it through, then we enter into a very important phase, which is called phase 3, and here we have thousands of patients that are tested for a year, and crucially, they're compared, the results of this test are compared to tests carried out with placebo and tests carried out with other existing treatments. So if you're looking at a new drug, you have to show two things. Is that the new drug is better than what we already had, and that new drug is better than placebo, because it's not just a placebo. Now, when we say it's better than a drug that we have, this has to be better in two ways, both in efficacy and toxicity, right? So it may not be as effective as the other drug in treating the disease, but if it's at least, if it has fewer side effects, if it's better orally available, if it's easier to administer it, if it's cheaper, all of that would be good, and it will probably lead to continuation with the use of this drug, and it will make it then to phase 4 clinical trials. Not all drugs have to go to phase 4, but phase 4 is the point when the medicine has been given marketing license, and the medicine is available on prescription, and so the efficacy and toxicity of this drug is now studied in a huge number of patients in all the countries where the drug has been licensed. But there will be a period of time where the monitoring of the drug will be very careful. So maybe you're aware that there were situations when some drugs will be licensed, and about 3 years later they will be withdrawn from use, because after having been available for a large number of patients, it became apparent that the drug, in fact, does have some very serious side effects, even though in a small number of patients, but the side effects might be so serious that they will have to withdraw it from the market. All right, so I thought I will tell you something about drug discovery process, because really this is what the whole point of translation is, right? So you're initially, we are scientists, we're basic scientists, we're in the lab, we work with various proteins, with various cell types, various tissues, various conditions, and we may come across some new and interesting function of a particular protein, say on the cell surface, or some of the metabolic molecules. And this would be the everyday life of many researchers in a pure research lab. Then once that occurred, we will enter into a position where, by identifying some target molecule, we would like to now design drugs that would affect that target molecule and modulate it in a disease state. So in this first part of the 3 to 6 years, we will carry out a drug discovery that is not only know which cellular molecule, which cellular target we want to attack, but we will employ a lot of chemistry or structural biology in order to devise or discover a specific drug-like candidate. And this is what we like to say is a drug discovery. And at that stage, in a typical drug discovery program, probably 5 to cellular molecules. And this may take 3 to 6 years, or even longer. So just going around, trying to come up with a special drug that you know interacts with whatever you want to interact, in a way you hope it does. And out of those, you will have a couple of hundreds of compounds that you may be using for some kind of preclinical trial. And then once that has happened, you will enter into clinical trials, and I told you about phase 1, phase 2, and phase 3. And this may take about another 6 to 7 years. And then finally, you will get approval, licensing, and it will go on the market, and they again may take up to 2 years. So there are two things that you will immediately see. One is that many years that are involved before the drug makes it to the market. So it's many, many years of work. And secondly, clearly, it's many years of work with many people, and hence, it's a lot of money as well. So the drug discovery process, it's really a collaborative research, and it involves many disciplines and expertise. So I didn't tell you what disciplines and expertise there are. But initially, you will probably looking into some cellular biologists, molecular biologists, structural biologists, physiologists, and so on, working together. And then chemists come along, quite a few chemists working really, really hard. And then there are other people involved, such that people studying toxicology, carry out toxicology studies. There are people involved just in commercialization process, in making this drug more commercially viable. There are pharmacologists, you name it. So it's a very large number of people, not to mention all the medics that are involved in actually, at the end, administering and testing these compounds in patients. The process, as I said, begins with understanding of the disease and identification of the human molecule or molecules that need to be modulated. And this is what we refer to as target identification and target validation. So we first identify molecular target in the cell, and then we have to validate it in some fashion. And from that, we carry on the process, but this is a key stepping stone. The process of drug discovery is also characterized by very high attrition rates, and that is we start from so many compounds and we end up with very few at the very end, but also not all of the targets that we were initially engaged in working towards designing drugs for them will be successful. So all of the drug companies would have many molecular targets that they're aiming for development of the new drugs, but at the end they may only come up with 1 or 2 drugs after the very long process. And I also already mentioned that the process is very expensive. How expensive is, in terms of dollars, the rumor is that for each drug to develop, you're looking into excess of $1 billion. Okay? So it's $1 billion for each drug to be developed. Now there is a lot of debate as to why this costs that much, or should it really cost that much, and can we make this cheaper, and so on. There's also a line of thought that of course it's going to cost that much, because then the pharmaceutical industry can kind of claim that they have to charge so much in order to recover their costs. But even though it's very expensive to develop a drug, one shouldn't forget that the returns are also very, very high. So for example, amongst 15 top-selling drugs in the last couple of years, each one of them had annual sales of about $5 to $10 billion. So that just tells you. And what do drugs look like? So I've shown you just a couple of examples to give you an idea. There are, in a way, 2 types of drugs that we are looking at nowadays. I would imagine you're more used to having drugs that drugs look like this. Some kind of cyclic compounds stuck together, and when you open if you do that, I do it, I don't know. Anytime I have a packet of some medicine, I open and I read and I try to look it up and I try to see what it looks like. Oh yeah, I'll remember this. And they all look the same to me. You know, it's just like a bunch of circles stuck together and you're wondering, how come this is a drug for A, while the other drug looking the same is a drug for B? And so I think because they all look in the first instance the same, simply because they need to have specific properties in order to be able to interact with proteins, with your target molecules. But in fact, if you were to look at these drugs in three-dimensional space, so if you were to look at them in a way how they're effectively structured at the point of interaction with the protein, you will realize that not only that they have significantly different structure, but also the distribution of these reactive groups, and they will have some polar hydroxyl groups or some chlorides and fluoride and so on, so that they like this sulfur in the ring or nitrogen in the ring. All of these interactive groups, the distribution of these groups will be such that allows for the specific interaction with the protein molecules at the target binding site that we were looking at. So that's commonly what we expect when we say drugs. And there is a whole science associated with what makes a good drug. Hopefully some of you will end up learning and being engaged in this, but there is a lot of work done as to what makes a good drug, how you can predict what size it should be, what kind of polarity it should have, and so on. And then here is a different drug. Okay, so this is a Lipitor. This is a cholesterol lowering drug, very, very well known. Majority of households will have them by now. This is Plavix, clopidogrel, very frequently administered drug for what do we say, lowering platelets settling, and this is commonly administered to heart patients and people post heart attacks. And this is a very different drug. I don't know how familiar you are with this, but this is a ribbon representation of the protein molecule. And here we have a complex between the FAB fragment, antibody fragment, against a specific cellular receptor. And this drug is called Herceptin. It's a very well-known and marketed drug that is used for treatment of breast cancer. It's a very different type of effect, very different type of interaction, but both of these are successful drugs, successfully used on the market. I'm stressing this because even though for many years this was what we were anticipating for drugs to look like, the use of monoclonal antibodies nowadays is really, really widely spread for cancer in particular. So I just wanted to mention something about the whole industry because frequently I come across a situation where I have to defend the drug discovery process and the pharmaceutical industry. And this is because people out there, some people, many people believe that the whole industry is primarily driven by profit and by greed. And this myth exists to the point that there is some population believing that the new diseases are invented so that the pharmaceutical companies would have something to work on and be able to sell as drugs. And I would really, really like to assure you that is not the case. That is not a primary reason why any basic science researchers enter into doing the basic science. To say that the pharmaceutical industry is not driven by profit is really wrong. Obviously, it's a business, and to a great extent, they will be driven by profit. But the whole process is something that you think it might help people. You can't help yourself, you just want to do it. And so you really, as a person, as a scientist, will do everything to find somebody who will give you money to push this forward to see whether you can do it. Can you make difference? Is that the question?
Yeah.
Yes.
with regard to that, as in, like thinking about the massive cost that goes into it, like a project might initially be started because of curiosity or philanthropy, but doesn't it lead to people sort of like pushing through projects because of the initial cost that they've suffered? Like if they've already paid £1 billion, they kind of have to make it.
Yeah, no. So this is where you're getting wrong. Nobody's paid £1 billion. Nobody is paid money to begin with. The £1 billion that I was referring to is a total cost to industry, and that means is paying the salaries of the people that are working in the process. Now, and this is why I wanted to come to this, is because when I start to work, I work without any money from drug companies. I work very hard trying to get funding from the government to allow me to work on the basic biology of some health or diseased state of human. Has nothing to do with being paid. At no point that I'm paid. Even if me or any of my colleagues get a £1 million grant from the government, none of it gets into my pocket. My salary does not depend on it. I do not get paid. What do I do with £1 million? I employ 4 people to work on the project for 3 to 5 years, where they're going to be making the protein, studying it using biophysical techniques, doing X-ray crystallography and mass spectroscopy, chemistry and so on. Right? I pay for chemicals, for the reagents, I pay for the use of facilities. At no point did I personally get any money. Does that answer your question? Thank you. When can I get the money? Okay, just to tell you. If, at that point, I and my colleague chemists say, I know how to target that specific pocket so that this receptor no longer works with that growth hormone. It can no longer be stimulated with that growth hormone, and we don't want it to be stimulated by that growth hormone because this would lead to cancer progression and angiogenesis. So at that point, we would say, we know how to target. So we make a molecule, we then patent that molecule, we then get a collaboration with the industry which is interested in investing further money to develop that, to employing 100 more people that would allow us to do all the studies associated with getting the potential drug to the market. And then eventually, if this drug does 10 and 15 years down the road make down to the market, I may get a small percentage of something if my rights were fully protected and if there was no dilution of assets on the way as an inventor that frequently inventors do happen to have because of the new investments coming through. Most of the money, when it comes through private investment at that point, so you as an inventor do not necessarily gain. You have a tiny little percentage, if at all. Yeah? So I will just quickly actually take you through one of the projects that I'm doing. I just want to give you this as an example. So we were working on small molecule inhibitors of neuropilin 1 and VEGF-A interaction. So just give you this in plain words. VEGF-A is vascular endothelial growth factor. It's the main growth factor that is really produced a lot in cancer, and what it does, it is at the moment we believe really important for growth of vasculature, which is needed for cancers to grow. So you need to deliver the blood to tissue, and therefore you need to make vasculature, and therefore you need a lot of VEGF. So this VEGF growth factor has to work on the cell somehow, so the VEGF will then bind to the cell, and then a signaling process will take place that eventually would lead to vascularization. The people that are involved in this work are many. So these are the people at UCL, we had the friends at Medical Research Council laboratory that were engaged. ARC Therapeutics was a kind of small to medium biotech company that funded initial parts of the applied part of the research, and then we did a lot of chemistry with another company called DomainX. I should tell you that initially DomainX and ARC Therapeutics were spun out from UCL. I'm giving you this as an example of new developments where we have a lot of academia working with industry or generating new companies from the academic research. So as I said, you have this vascular, you have this, you have a growth factor, VGF, and this will interact with proteins on the surface of the cell, and it will interact with this VGF receptor as well as with this receptor called neuropilin. What we wanted to do is to interfere with interaction between VGF and neuropilin at this site, and basically screw up their business. You know, basically say, okay, this no longer is functioning receptor ligand process. So you first have to go through a process of target validation, and that effectively means, you know, is this expressed in tumors? If you use antibody against it, will the tumors stop growing? Does it work in combination with known therapies and so on? So I will just show you that we did what we call two-pronged approach, and that is looking at biological space of that interaction and chemical space. So we know that the ligand and the receptor interact. So first we wanted to know what is important within that binding site. Which specific amino acids within that binding site are important for that interaction. And we do mutagenesis, we mutate individual amino acids and look for the effect. This is what we call probing biological space. And then we make molecules that are that are similar to the ligand, and change those molecules in many ways to see which molecule fits better, and that's what is referred to as probing chemical space. So this is the results of biological space and chemical space. It's just a prototype molecule at the end. Doesn't it look just like the other drugs I've shown you? Yeah, exactly. So at the end we came up with yet another. You know, here are some circles, here are some hydroxyl groups, here are some nitrogens, and so on. Okay. I'm a structural biologist, so I was particularly involved in deriving very detailed picture of interaction between that drug molecule and neuropilin. We were focusing on this B1 domain of neuropilin and solving various structures. We also used NMR spectroscopy to map out the surface of interaction. And so at the end we were able to derive not only beautiful pictures like this, but also very detailed understanding of the level of interactions at atomic level, and then and get ideas how we can then change the drug to make it more potent, more orally available, better clearance to the body, and so on. In summary, what we did, we used molecular modeling, we used NMR spectroscopy, X-ray crystallography, surface plasma resonance, isothermal calorimetry, all of these biophysical techniques to start from just knowing what the original biological ligand looks like, VGF, you know, this vascular endothelial growth factor, and then slowly go from making active peptides, then carry out some what we call structure-activity relationship of active peptides, do crystallography, and get more and more understanding of both chemistry and biology, and then we would come up with what we call our prototype EG229 peptide-dolimetric drug. And we are still developing now. Where we are now, we now have another new company called Magnus Life Sciences, which is an umbrella company with 5 businesses, but 2 businesses, Magnus Life and Magnus Metabolic, are focusing on developing drugs targeting exactly this interaction, both in cancer and diabetes. And through this private funding, we now have secured commitment for funding at least up to the phase 2. We also have funding from British Heart Foundation and Medical Research Council to look further into the basic biology and structural biology of this neuropilin-VGF interaction. So I think this is a way forward to actually foster closer links between academia and industry. And through this, we can encourage innovation, right? Knowing that you're doing basic research, but there is a formalized fashion. This would enable development of new treatments which are grounded in basic research, which are carried out by leading researchers. More importantly, now coming back to profits and profit-driven businesses. By integrating basic research within academia and industry, we can afford to tackle rare and less profitable diseases. Okay? So we can afford to actually tackle those diseases that within the environment where the profits are primary aim, we should be able, they may be neglected. So believe me or not, antibiotics have been neglected for the last 20 years because the drug companies decided that they were not sufficiently profitable. And now we're coming to a situation that we're really, really in a crisis, such that we need new type of antibiotics. And so a lot of basic research is carried out in development of new ways of treating bacterial infection. And by doing this basic research very thoroughly in academia, we will be able to eliminate risks earlier on in order to reduce attrition rates and costs of drug development. So that's all I had to say. If you have any more questions about this, I'm happy to answer. I also know that some of you are interested in knowing something about the application process to UCL. So again, I'll be able to answer some questions on that if you're interested. Yes.
You know.
I think you're right when you talk about curiosity in terms of the development stage of the research of drugs. In terms of actually producing them and selling them on an industrial level, I think profit does, in terms of businesses, become a more primary motive than it was before.
Right. But that's exactly why it's good if you start from academic environment, you carry out a thorough research, and you develop a right drug. Sure enough, you can then influence, and of course the pharmacist will have to be, it's a business, right? It's not a charity organization. So if you produce a right drug, you will at least, I was trying to explain, you will at least lower down the costs such that selling the drug would be cheaper.
But my question on that, thanks for explaining that, is that do you think currently, because of what you just said, there exist drugs which treat more less profitable diseases, the cancers, the HIVs, but since they're not economically viable, we haven't seen them in production?
No. Okay, the short answer is no. Again, that's another myth, that there is somebody has out there found a drug for cancer, they just don't want to tell us about it because it will ruin their business of selling other drugs, right?
Well, they don't go into research that much because they don't at the moment see a client where it could sell for as much money as, say, another drug which is more commercial.
But the research is not driven by pharmaceutical industry so much. Really, the main research is driven by academia and research institutes. You don't grow up to be a scientist so that you will say, I'm going to go to pharma to do research. If you want to be scientists and do research, you do not go right away to pharma. At some later stage, you may decide that that's what you want to do. But what you want to do is have your own lab and do research in whatever you want to do. And you don't want to go to pharma exactly for that reason, because when you go to pharma, you'll have to do research in what you're told. So all the major discoveries are really done primarily within academic environment, where I mean universities and research institutes. Yes, again.
To what extent does like a researcher like yourself remain part of the development process?
I'm sorry, I didn't hear that. To what extent?
Does a researcher like yourself remain part of the development process, or is there any point where you have to like sort of back out of it?
No, I don't need to back out of it. I mean, at the point when my expertise are no longer needed, I would say. So if they're going to say, yes, this is a drug we're going to make, and now it's going to be going to the process of formulation. Okay, what does the formulation mean? It means, when you take a tablet, it's not just the drug. There are all other things mixed in together so that you can ingest it properly, so that it can be dissolved appropriately, so, all of that. So I know nothing about formulation. So at that point, I'm no longer needed. Does that make sense? So I'm not picking up. It's just I've done my job. Now it's time for somebody else. I don't know how to administer drug to patients. Obviously, that would be something that medics would do. Anything else? Oh, yes.
You mentioned that sometimes the government funds some research trials.
Some research?
Some research is government funded.
Or a majority of research is government funded.
Does the government ever pull out of that kind of research and tell us to stop?
Pull out?
Yeah, in terms of funding.
All right, I'm not sure that I understand your question 100%, but what I would explain is that the government has research councils, which for many, many years operate in the same fashion, and that is they have regular opportunities throughout the year when researchers can apply for funding. And there are many different ways of applying, or many different types of funding that can be obtained. Some that are 3 years, it's called just basic major project funding, or program grants which go for 5 or sometimes even a little bit longer years. Most of the times these calls are very broadly open. You can apply to do the research in whatever you want. But for the most of the time, the research councils will have their general strategic goals. So if you can fit into their strategic goals, perhaps the chances of obtaining funding are somewhat better. But otherwise, they funded the research that does not necessarily fit into their strategic goal as well. It's all about just how well you can convince them that you are the right person to do that work, and how well you thought about it, and so on. And the success rates are, say, up to 20%. So every time you submit a project grant, there is a chance of 20% of you getting it. Once the funding is approved, nobody will say, oh, I'm sorry, you no longer have money. Basically, once the funding is approved, the money is transferred, and that's it. You do have to report on it and to justify, obviously. You can't just say, and spend it, there's still accountability going on. And if you don't spend it well, next time you ask for it, you're not going to get it. But the government budgets are adjusted, and sometimes they may cut down the amount of money that comes to the pot. And so, for example, this year the government is not cutting down. They're maintaining their budget, or about. That effectively means reduction in funds because of the inflation. Yeah? Yes?
Do you think that there are some diseases or some illnesses out there that are curable, or drugs can be made for them, but drugs simply aren't because there isn't enough demand for them?
Yeah, so this is what I was referring to earlier, that by having this mechanism, we might be able to tackle more of rare diseases, those that are less profitable. And my colleague, Professor Elizabeth Shepherd, for example, now has kind of industrial collaboration with somebody else, which is partially funded by Medical Research Council, to tackle very rare condition, which is called fish odor syndrome. Okay? It's not life-threatening disease, but people that have this syndrome have metabolic disorder, which makes them smell like rotten fish. It sounds funny in the first instance, but for people that do have this condition, it's really not laughing matter, because obviously nobody wants to sit around them, okay? And so in order to help the lifestyle of these people, it's very hard to convince, big pharma, let's make drugs against fish odor syndrome. However, there is a need for this disease, and she's now working on developing a treatment for that. Yes.
Which and how many drugs have you been involved in the development for?
Which and how many drugs?
Yeah.
Okay, so I'm specifically involved in development of these 2 series of drugs, one for treatment of diabetes, another one for melanoma. I also have a collaboration which is at much earlier stages for treatment of post-heart attacks and preventive cardiovascular treatments. So I'd say that's about it. He hasn't asked me.
Hi, do you think that big pharma places...
The reality is, as I said, pharma is a business. So yeah, they will do everything to protect their business. But at no point the big pharma is going to come down, knocking your door and say, sign a contract with us, and you will say, no, I would rather do it by myself. You can't do it by yourself. you don't have the money, you don't have the skills, you don't have capacity, you need to be backed by funding.
But that gives all the power to Big Pharma then?
No, because you have a know-how. You know how to do it, they're just interested in paying for it.
Right, but if you know how to do it and you don't want to accept funding,
You can't do it.
then you can't do it.
Exactly.
So that gives all the power to Big Pharma.
No, not necessarily. Because it's not only one pharmaceutical company,
Okay.
it's not only one, there are theoretically situations where you can, okay, Bill Gates Foundation, for example, funds development in malaria target treatments. He's not the pharmaceutical industry. Yes.
This question's not on funding.
It's not on funding, okay.
In terms of clinical trials,
Yes.
how do you overcome the ethical implications of using humans as a subject?
Right. Well, I think that's an easy ethical implication. It's far easier than saying, you know, you have to use humans as subjects because you want to treat humans, right? So if you were to use humans as subjects for treatment of veterinary diseases, I think that would be a problem, and it wouldn't be right. But you must use humans because you cannot translate all the findings, say in rodents or so on, to human physiology. It's clearly difficult, and that's why you initially start with healthy volunteers that would, and it's a small cohort, and you just have to test for the toxicity. Before you go into humans, you go through rats and rabbits and all kind of other animals that you can do, but there's only how far you can get with the animals. So obviously you will treat animals first. You're not going to go to humans directly from the cell culture study. Yeah. There are ethical issues, obviously, because this is again something you don't want to go. I personally, thankfully, do not have to think about it, but you can hear the rumors of some drugs being tested or thought to be tested in less developed countries and so on. Clearly, that's not something you want to be engaged in, right? Yes.
To what extent is there competition from other researchers if they're researching the same drug or the solution for the same?
Yes. So this is a required component of any application for any funding. That in order for either government to contribute to or to get a private investment to your project, you really have to convince them that either there isn't a competition, that you're fully aware that you're that you're so early on and that you're so advanced that there is no legitimate competition. But also, if there is a competition, that you need to show them that you are aware of competition and where they are at, that you know what's happening, and you need to convince them that your approach is superior. If there is any doubt, you won't get money. Again, coming back to funding. Yeah. Yes.
In the first wave of human trials, when you look at testing on healthy volunteers,
Yes.
how often does that, go
Goes bad?
really bad?
Yes. I didn't have time, but there are occasions when it goes terribly bad. And I think the most famous one is from back in 2006. I don't know if you're too young to remember, but there was a trial for some antibodies to interfere with the immune response and so on. They were really, really, really badly designed trials. Normally you start with very small doses and so on, and they're very controlled. In this case, there were, I believe, only six patients, and they had developed really, really severe reaction. One of the patients, one of the people, his head was completely swollen and deformed and so on. So it was a massive scandal. This is this drug called TGN-14 or something like that. You can look it up. So I think every once in a while there is evidence that things have gone very badly wrong. But this is the only one where I could say terribly wrong things went terribly wrong that I'm aware of, and as of lately. The fact that things went wrong with that particular one really made regulations tightening even more. So there is no one who wishes to repeat that experience.